Okay, so today we will talk about hematopoiesis. In the previous lecture, we discussed red blood cells, white blood cells, and platelets, and we looked at the functions of each of them and some, classifications also of each of the blood cells. But today we're going to look at how they are produced. We had mentioned before that red blood cells are produced through a process that is called erythropoiesis, and white blood cells are produced through a process called leukopoiesis, and then platelets are produced through a process called thrombopoiesis. But now, today we would look at hematopoiesis, which is the process of the production of each of these blood cells. So we'll see how red blood cells are produced, the whole process, how white blood cells are produced, and how platelets are produced. Now, this topic is very important because if you don't understand or you don't know the process of production of the blood cells, you might not be able to diagnose some clinical conditions like leukemia in the future, aplastic anemia, and all of those things. You will not be able to diagnose them. But if you know the process of production, you would know the difference, for example, between acute and chronic leukemias. You would know the difference between, aplastic anemia and other types of anemia, because in your exams, you're going to get questions where they would give you the number of each of the blood cells. And you need to know if these blood cells are excessively increased or if they are decreased. And if they are decreased, why is it decreased? What happened? Was there a problem with the process of production? So you need to know all of that. So that is why this topic is very important. So let's get started. Hematopoiesis. So hematopoiesis, from the word you could see hem is blood, or hemato, you could see blood, while poiesis is the process of production. So hematopoiesis simply means the production or the process of production of the different blood cells. Now, in this lecture, we would define what hematopoiesis is. We will talk about the anatomy of hematopoiesis, and we would talk about the cellular pathways of hematopoiesis, the specific differentiation pathways, and hematopoietic growth factors, and finally we'll talk about the formation of blood cells. Now, here you would find that we'll have definition, we'll have the anatomy. I'm going to give you a diagram, that shows you in details how each of these blood cells are produced, and then we'll look at the different pathways, and we'll look at specific differentiations to produce specific cells. And then we'll look at the growth factors, which I mentioned some of them in the previous lectures. I talked about erythropoiesis, I talked about thrombopoiesis and leukopoiesis. We'll see them here again. And then we'll look at the formation of the blood cells. So let's start with the definition of hematopoiesis. So hematopoiesis is simply the production of red blood cells, white blood cells, and platelets. Hematopoiesis is the process of production of red blood cells, white blood cells, and platelets, or it is the process of blood cell formation. We had said earlier that the blood is made up of the blood is made up of the plasma, right, and the cellular elements. So now we are talking about the production of the cellular elements. Why? Because the plasma, we already said, is made up of water, mainly 91.5% water, and then we have proteins and ions and mineral elements. But the cellular elements themselves are not made from water. So the cellular elements are made by a process called hematopoiesis. which is what we're talking about today. Now, so what is the anatomy of hematopoiesis? How does it start? Where does it start? And when does it start? Now, in the first trimester of pregnancy, when you still have a fetus in the womb, in the first trimester of pregnancy, that is to say, maybe the first 4 months of pregnancy, the first 3 months of pregnancy, the process of production of blood cells takes place in the yolk sac. In the yolk sac, that is where hematopoiesis takes place. When the fetus is between the first month and the 3rd month of pregnancy, or the first trimester of pregnancy, we have hematopoiesis taking place in the yolk sac. And then in the 2nd trimester of pregnancy, between the 3rd month or the 4th month, so between the 3rd month and the 6th month of pregnancy, we have hematopoiesis now taking place in the liver and in the spleen. Now, we're talking about the liver and the spleen of the fetus. That is where hematopoiesis takes place. And then in the 3rd trimester of pregnancy, from the 6th to the 9th month of pregnancy, where the baby now has the fetus now has some bones, yeah? So hematopoiesis now takes place in the central and peripheral skeleton. So now the baby or the fetus has some bones now. So that is where hematopoiesis takes place. But after childbirth, when the baby is now born and the child grows to become an adult, so as an adult, hematopoiesis takes place in the axial skeleton. So we have the vertebral bodies. You have your vertebral column, and they have each of the vertebrae. They have bodies, vertebral bodies. So hematopoiesis takes place in the vertebral bodies, in the sternum, the breastbone, and then in the ribs and in the pelvis. So these are the places, the bones, where hematopoiesis takes place. Now, I had said in the previous lesson that in a fetus, hematopoiesis takes place in the liver and in the spleen. In an adult, it takes place in the bone marrow. Okay, that was what we said in the previous class, which is what we're seeing here now. So in an adult, hematopoiesis takes place in the skeleton or in the bones, especially the vertebral bodies, the sternum, the ribs, and the pelvic bones. Now, hematopoiesis may expand into fetal sites in times of severe demand, e.g. thalassemia. So when there is a severe demand for the production of blood cells, let's say, for example, somebody is having maybe sickle cell anemia, where there is shortage of red blood cells, or there is thalassemia, where there is also a shortage of blood cells with abnormal shapes in thalassemia. At that time, your body needs more blood cells. So what does your body do? The bone marrows alone are not enough, or the skeletons alone are not enough to produce the amount of blood cells that are needed at that time. Why? Because that is the time of crisis. There is severe demand. The person is suffering from thalassemia or maybe sickle cell anemia. At that time, hematopoiesis may re-expand into fetal sites. So what happens is that at that time, the liver and the spleen might want to produce blood cells also. So at that time, not only the skeletons will be producing blood cells. Hematopoiesis will not only take place in the bones, it will also take place in the liver and in the spleen. And we call that extramedullary hematopoiesis, because we had said that the normal hematopoiesis in an adult takes place in the bone marrow, okay? But when there is a high demand or a severe demand in times of thalassemic crisis or anemic crisis, what we have would be an extramedullary hematopoiesis, because some blood cells will be produced in the liver and in the spleen, which they are not supposed to, where they are not supposed to be produced on a normal day. So that is what we call extramedullary hematopoiesis. So don't forget that. You could be asked in an exam, what is extramedullary hematopoiesis? So it simply means the production of blood cells in areas that are not the bone marrow in an adult. So we have production of blood cells outside the normal bone or the normal skeleton where they are produced. So they could be produced in the liver or in the spleen, which were the locations for production when the baby was still in the womb as a fetus. Okay, so that's what we call extramedullary hematopoiesis. So now let's go to the cellular pathway of hematopoiesis. Now we're talking about cells, and all these cells we're talking about are red blood cells, white blood cells, platelets. Yes, so all of these cells, they come from different pathways, and we're going to look at the pathways where they are coming from. Now, the overall cellular organization of hematopoiesis. Hematopoiesis begins from what we call stem cells. So the stem cells are the first cells from which the process of hematopoiesis starts. So stem cells are of two types. We have the totipotential cells, or what we call totipotent cells. So we have totipotent cells, and these totipotent cells further give rise to what we call multipotential cells or multipotent cells. I'm going to explain what I mean by totipotent and multipotent. You will see in the next slide that totipotent cells are cells that have unlimited ability. They have unlimited potential to divide into different kind of cells or to differentiate into different kind of cells. We'll look at that later. But know that all blood cells that you see are produced from stem cells. So the first cells involved in hematopoiesis are called stem cells. And these stem cells are of two types: the totipotent cells with unlimited abilities or unlimited potential to differentiate. And then these totipotent cells divide and give rise to what we call multipotent cells. And then after the stem cells, the second group of cells we have are called the progenitor cells. So the progenitor cells are formed from the stem cells or from the multipotent cells and the totipotent cells. So we have the progenitor cells. And then these progenitor cells have, a kind of limited ability to differentiate. We're going to look at that. And then these progenitor cells give rise to precursor cells. And then precursor cells give rise to effector cells. So when you see a red blood cell, for example, a red blood cell will be a kind of effector cells. This is the final kind of cell that is produced. Red blood cells, white blood cells, platelets are the final cells, effector cells. So these effector cells were produced from precursor cells. And these precursor cells were produced from progenitor cells. And then these progenitor cells were produced from stem cells. Now, it means that all blood cells go through this process of production. Now let's go to the next slide. So what are stem cells? Let's look at stem cells. So stem cells are, as we said, are the originators or the first group of cells that initiate the process of hematopoiesis. Okay, they are the foundation from which all other cells are formed. Now, these stem cells, we had said that are of two types: the totipotent stem cells and the multipotent ones. So for the totipotent stem cells, they are also called the godfather cells. Why? Because we had said that they are the first cells from which all other cells are produced. So totipotent cells is a type of stem cell, okay? Now, what are the characteristics of the totipotent stem cells? They have unlimited self-renewal. So it means that one cell that is totipotent can renew itself. It means it can divide into two. So I have one totipotent stem cell. That cell can divide into two cells. It can renew itself. That's what it means. It can reproduce itself. And it has unlimited abilities to do that. So a stem cell or a totipotent stem cell has what we call unlimited self-renewal. It can replace or renew itself. It can divide to form another kind of itself. Okay, so it's like you have one orange splitting into two different complete orange, not half-half, but two different complete orange. And then you have each of these two different complete oranges splitting again to form, another two different, oranges that are complete, not half-half. So unlimited self-renewal. Now, apart from unlimited self-renewal, they also have what we call unlimited differentiation. What does that mean? It means that totipotent stem cells can give rise to any cellular element. So a totipotent stem cell can decide to give rise to an erythrocyte, or it can decide to give rise to a leukocyte, or it can decide to differentiate into a thrombocyte. So totipotent stem cells, they have unlimited differentiation. They can differentiate into any kind of cell that they want. They can either give rise to erythrocytes, or they give rise to leukocytes, or they give rise to thrombocytes. So that is differentiation. And they have unlimited abilities to do that. But they are present in the bone marrow in small numbers. They are not a lot. They are very small numbers of them in the bone marrow, because, of course, they have the ability to differentiate into any kind of cell, and they can reproduce themselves if they want. So they are only in small numbers. But whenever the need arises, they can split, okay, and multiply themselves if they need to. Then they are highly resistant to chemotherapy. So when they cannot be easily destroyed by chemotherapy. So when somebody has, for example, cancer, right, and you want to treat with chemotherapy, most times those chemotherapy or the chemicals that are used are able to destroy some cells, some blood cells. But these totipotent stem cells, they are highly resistant to chemotherapy. And then they reside primarily in the bone marrow. Why? Because that is where the hematopoiesis takes place. Only very small number of them circulate in the blood. So remember that. They reside primarily in the bone marrow. And then for them to differentiate, they need an antigen, a surface antigen, which is called CD434+. So CD means cluster of differentiation. Okay, we will do that in immunology later. So the totipotent stem cells, they have a surface antigen, which is called CD4+, CD34+. This could be an objective question in an exam. So remember that totipotent stem cells have a surface antigen that is called CD34+. Sorry, CD4. I'm talking about CD4 so much because of HIV. Okay, so, and these totipotent stem cells, they look like small lymphocytes. We had a class about lymphocytes, a lecture about lymphocytes in the previous class we had. So these totipotent cells, they look like small lymphocytes. And then if there is a dysfunction in totipotent stem cells, it will lead to aplastic anemia or certain types of leukemia. Remember that we said that all blood cells are produced from stem cells, and totipotent stem cells are examples or types of stem cells. So if there is a dysfunction of totipotent stem cells, we'll have what we call aplastic anemia. In aplastic anemia, there is absence or very decreased number of red blood cells, white blood cells, and platelets. So you wouldn't find, we already mentioned, for example, that we have about, we talked about 4.8 to 5.4 million red blood cells per microliter of blood. But imagine that you take a microliter of blood and you cannot find even, 10,000 red blood cells. or you cannot find red blood cells at all, then we know that there was no hematopoiesis. There was no hematopoiesis. So we call that aplastic anemia. There was no production at all of red blood cells. And if you also cannot find the white blood cells, then it means that there was no production of white blood cells. And why? Because the problem was with the stem cell. Remember, it is the stem cell that gives rise to all cellular elements. It is a stem cell. So if there is a problem with the differentiation of stem cells, we would not have, you know, any blood cell that was supposed to be produced. So that's, we will now have a kind of anemia, which is called aplastic anemia, or we could have some certain types of leukemia if the totipotent stem cell did not properly differentiate or give rise to multipotent stem cells, then we could have some certain types of leukemia. Okay, now let's look at multipotent stem cells. So multipotent stem cells are of two types: lymphoid and myeloid stem cells. Lymphoid and myeloid stem cells. Now, for totipotent stem cells, we say that they can differentiate into any kind of cell they want. But for the multipotent stem cells, look at what happened. The first thing you need to know about multipotent stem cells is that they are derived from totipotent stem cells. So it means that if there was a problem with the differentiation of totipotent stem cells, we will not have multipotent stem cells. And if we don't have multipotent stem cells, it means we will not have the lymphoid and the myeloid stem cells. And if we don't have lymphoid stem cells, we cannot have lymphocytes. If we don't have myeloid stem cells, we cannot have erythrocytes, we cannot have thrombocytes, we cannot have granulocytes, neutrophil, eosinophil, and basophil. We cannot have them if the totipotent stem cells could not give rise to multipotent stem cells. Now, so multipotent stem cells come from totipotent stem cells. Now, they are capable of extended self-renewal, but not unlimited, as we said about totipotent stem cells. But they are capable of extended self-renewal. They can renew themselves to an extent, you know, okay, but not unlimited. And then they are also capable of extended differentiation, but not unlimited differentiation. Why? Because we said that totipotent stem cells can differentiate into any kind of cell they want. But multipotent stem cells, they have multiple potential to differentiate, but not unlimited. They cannot just differentiate into anything they like. Multipotent stem cells can only differentiate, one, into, we have what we call the lymphoid stem cells. And the lymphoid stem cells will give rise to lymphocytes. So when multipotent stem cells differentiate, they either differentiate into lymphoid stem cells or they differentiate into myeloid stem cells. They cannot differentiate into all kinds of cells. That is why they are not unlimited. They don't have unlimited differentiation. They only have an extended differentiation. For lymphoid stem cells, they can give rise to all categories of mature lymphocytes. So it means that they can give rise to T lymphocyte, B lymphocyte, and they can give rise to helper T lymphocyte, suppressor T lymphocyte, and killer T lymphocyte, but only lymphocytes. Okay, so the lymphoid multipotent stem cells can only give rise to lymphocytes. While the myeloid stem cells, now pay attention to this one, very important. The myeloid stem cells give rise to red blood cells, granulocytes, that is neutrophil, eosinophil, and basophil, monocytes, and platelets. Now, it means that the myeloid stem cells give rise to more cells, more cell lineage than the lymphoid stem cell, because lymphoid stem cells can only give rise to lymphocytes. But myeloid stem cells will give rise to red blood cells, So type of white blood cells: neutrophil, eosinophil, and basophil; monocyte, which is also a type of white blood cells; and then platelets. So it means that if there is a problem with this myeloid stem cell, we would have problem with our red blood cells, with our white blood cells, except the lymphocytes, with monocytes, and with platelets. This is important because when you start talking about different diseases that arise from red blood cells, white blood cells, and platelets, you need to know where the problem is. If the problem is with the myeloid stem cells, so you should expect that it will affect all these group of cells. All this group of cells will be affected. But if the problem was with the lymphoid stem cell, then only lymphocytes will be affected. So if you have lymphocytopenia, decreased number of lymphocytes, then you know that maybe there is a problem with the lymphoid stem cells. Or if you have excess lymphocytosis, pathological increase in the number of lymphocytes, for example, you know that there's a problem with the lymphoid stem cells. Now, number 4, the fourth thing you need to know: lymphoid stem cells can give rise to so-called lymphoproliferative malignancies. So now this is a kind of cancer of the white blood cell, of the lymphocyte, which is called acute lymphocytic leukemia. So in lymphoproliferative malignancies, there is excessive proliferation of lymphocytes. So you see the lymphoid stem cells giving rise to excess lymphocyte. There is proliferation of the lymphocyte in excessive number. And we had said before that instead of the normal amount of lymphocytes, we said it's 30% of the white blood cells in our last lecture. So imagine that we now have lymphocytes that are 70% of the white blood cells. That would be it will show that there is an excess proliferation, or what we call lymphoproliferative malignancy. So it proof that there is a kind of leukemia of the blood, or cancer of the white blood cell. And then the myeloid stem cell can also give rise to what we call myeloproliferative malignancies, such as the acute myeloid leukemia. So this leukemia also is caused by excessive proliferation of the myeloid stem cells. So anything that makes lymphoid stem cells or myeloid stem cells overproliferate, you will have leukemia. Either it is acute lymphocytic leukemia, where there is excess lymphocyte production with the presence of what we call blast cells, which we'll talk about later. Or you have acute myeloid leukemia, where there is excess production of myelocytes with the presence of blast cells, myeloblast here and lymphoblast here. We'll talk more about them later when we're discussing leukemias. So, but just know that any excessive proliferation of multipotent stem cells can lead to leukemia. Now, we said that after totipotent stem cells, we have multipotent stem cells. And then after multipotent stem cells, we have what we call progenitor cells. So totipotent stem cells will give rise to multipotent stem cells, and then multipotent stem cells will now give rise to what we call progenitor cells. Now, these progenitor cells, we also call them the colony-forming units. The colony-forming units. Now, they are derived from the multipotent stem cells. They are capable of limited self-renewal, and they are capable of limited differentiation. So at this point, the cells can only differentiate in a limited fashion. It means that they cannot differentiate into all kind of cells, and they cannot differentiate even into all myeloid cells. Let's take an example. Here we have what we call colony-forming units, granulocytes, erythrocytes, macrophage, and megakaryocytes. So what does it mean? It means that this type of progenitor cell, CFU, GEMM, can only differentiate into granulocytes, into erythrocytes, into macrophage, and into megakaryocyte, this one. Then we have what we call the colony forming units, megakaryocyte and erythrocyte. So this one now, notice that it can only differentiate into two things. One, megakaryocyte, which will give rise to the platelets, and then the erythrocytes. This colony forming unit, this progenitor cell, cannot differentiate to form a granulocyte. It cannot form neutrophil, it cannot form basophil, it cannot give rise to eosinophil. It can only give rise to platelets from megakaryocytes, and it can give rise to erythrocytes. That is why we say they have limited differentiation. It cannot decide to differentiate into lymphocytes, for example. It cannot do it. But totipotent cells could become lymphocytes. They could become erythrocytes. They could become thrombocytes. Multipotent cells could become lymphocytes. But multipotent cells cannot become both lymphocytes and myelocytes. They cannot. But now, when we get to progenitor cells can only have limited differentiation. So they can only differentiate into a particular or two, cells. For example, we have a colony forming unit, megakaryocyte and erythrocyte. These colony forming units can only form megakaryocytes or platelets, from which platelets are formed, and then erythrocytes. Then we have the colony forming unit, granulocytes and monocytes. This one now can only differentiate to form granulocytes, which are neutrophil, basophil, and eosinophil, and then can differentiate into monocytes also. But this one cannot differentiate into erythrocytes. You see the difference now. Here, colony forming unit GEMM can differentiate into any of these. But when it becomes this colony forming unit, it can only give rise to megakaryocytes and erythrocytes. This one can only differentiate into granulocytes and monocytes. And then colony forming unit G, which is granulocytes, can only form granulocytes. It cannot form monocytes, it cannot form erythrocytes. So it had limited differentiation. Now, if they ask you in an exam, which of the following progenitor cells give rise to erythrocytes? Which of the following progenitor cells give rise to erythrocytes? A, CFU-ME, B, CFU-GM, C, CFU-G. The answer would be what? CFU-ME, because that is the only one that can give rise to erythrocytes. If they say, for example, which of the following progenitor cells can give rise to megakaryocytes or can give rise to platelets, which of the following can give rise to platelets? A, CFU-ME, B, CFU-GM, C, CFU-G. The answer would be CFU-ME, because we said that megakaryocytes will give rise to platelets. So only these progenitor cells can give rise to this. So it means that if anything happens to these progenitor cells, CFU-ME, it will not affect granulocytes in the blood. You will still have normal granulocytes, but you might have decreased or increased platelets and erythrocytes. So there are some diseases of red blood cells, or there are some diseases of blood cells that you will find in the question they will give to you. You can see that there is excess erythrocytes and there is excess megakaryocytes or excess platelets. But granulocyte is normal, monocyte is normal, lymphocyte is normal. Then you know that the problem affected only these progenitor cells, because what we see in the blood now is excessive platelets and excessive erythrocyte. But every other blood cell is normal. That's to tell you that where the problem was with this cell, these progenitor cells. Okay, number 4. Now, these progenitor cells, they are responsive to hematopoietic growth factors. they are responsive to hematopoietic growth factors. So what are these hematopoietic growth factors? We mentioned some of them before. We talked about erythropoietin. We said it stimulates the production of red blood cells. So here we see erythropoietin is a hematopoietic growth factor. It stimulates the colony forming unit erythrocyte. So CFU, colony forming unit erythrocyte. This is the progenitor cell that give rise to erythrocytes or to red blood cells, and they respond to erythropoietin. Anytime erythropoietin is released, colony forming unit erythrocytes are stimulated to produce more erythrocytes. And then we have the GM CSF. So what does this mean? This is granulocyte, monocyte, colony stimulating factor. Granulocyte, monocyte, colony stimulating factor. Stimulates the CFUGM to produce granulocyte and monocyte, which is the second progenitor cell you see here. So this is the growth factor, GMCSF, granulocyte, monocyte, colony stimulating factor. It will stimulate the colony forming units, granulocyte, monocyte, to produce granulocyte and to produce monocyte. Then we have the GCSF, which is the granulocyte colony stimulating factor. We stimulate the colony forming unit granulocyte, which is this last progenitor cell here, okay, to produce granulocyte. Now, if we have, for example, we talked about that the production of white blood cell is stimulated by what we call leukopoietin. So you could call this one, because these are leukocytes, granulocyte is an example of a leukocyte, okay? Monocyte is an example of a leukocyte because they are white blood cells, yes? So we could call all these growth factors, you could call them leukopoietin, but for erythrocytes, it's called erythropoietin. Now, what growth factor will stimulate megakaryocyte production? So what will stimulate colony forming unit M, only M, not E. Imagine that we have colony forming unit megakaryocyte. What growth factor will stimulate it? Thrombopoietin. Because we said that colony forming unit megakaryocyte will form thrombocyte. So to stimulate that, thrombopoietin will be the growth factor that will stimulate it. Do you understand that?
Yes, sir.
Okay, good. Now, the progenitor cells, they express differentiation antigens, which are surface proteins, such as cluster of differentiation 19 B lymphocyte. We said for totipotent stem cells, they had a cluster of differentiation called CD34+, okay? But for these progenitor cells, they have CD19. And this particular cluster of differentiation will give rise to B lymphocyte formation. Okay, let's look at the next slide. So we said we have totipotent cells. They give rise to multipotent cells, and multipotent cells give rise to what? Progenitor cells, right? And then progenitor cells will now give rise to precursor cells. Let me take you back a little bit. So here we said stem cells, okay, or the totipotent cells give rise to multipotent cells. Multipotent cells give rise to progenitor cells, and these progenitor cells give rise to precursor cells. So we have looked at totipotent cells, they give rise to multipotent cells. Multipotent cells give rise to progenitor cells. Now progenitor cells will now give rise to precursor cells. So let's look at what the precursor cells are like. So these are precursor cells. Now precursor cells are blast cells and their progeny. Blast cells and their progeny. So blast cells, we would look at them later also. Now, before you get an erythrocyte, you must first get an erythroblast. So blast cells give rise to cytes. In the process of differentiation of blood cells, you first get a group of cells that are called blast cells. So it means, let me take you back a little. When this colony forming units, megakaryocyte, erythrocytes, when it differentiates, it will not give rise to erythrocytes directly. It will first give rise to erythroblasts before erythroblasts will now divide again to give rise to erythrocyte. The same thing will happen megakaryoblast also. If it's a lymphocyte, for example, you have a lymphoblast before you get a lymphocyte. Okay, so that is what we call the precursor cells. So the precursor cells are blast cells, and these blast cells are what you find in leukemia. One of the way you diagnose that somebody has a leukemia is the presence of blast cells in the blood, which are not supposed to be there. Because what we're supposed to see in the blood are cytes, erythrocytes, not erythroblasts. We're supposed to see lymphocyte, not lymphoblast. So anytime you see a blast cell in the blood, lymphoblast in the blood, it shows you that there is a problem with differentiation. It means that there is a leukemia somewhere, and the number of the blast cells will help you differentiate whether it is an acute leukemia or it is a chronic leukemia. But we'll talk about that in a future lecture. Now, so the first morphologically identifiable cells are the precursor cells. These are the first cells that you can morphologically identify, because all the previous cells we were talking, the colony forming unit we're talking about, you cannot actually identify them, morphologically. But the first morphological cells that you can identify are the precursor cells or the blast cells. So we'll have erythroblasts, which will now divide again or differentiate to give rise to red blood cells or erythrocytes. Then we have myeloblasts, which will give rise to granulocytes. So neutrophil, eosinophil, and basophil will come from myeloblast. And then we'll have monoblasts, which will not differentiate or divide to give rise to monocytes. Then lymphoblasts will divide to give rise to lymphocytes. And then megakaryoblast will not divide to give rise to platelets. So we see that the precursor cells give rise to the final cells, which are the red cells, granulocyte, monocyte, lymphocyte, platelet. So here we have red blood cell, white blood cell, white blood cell, white blood cell, and then platelets. They have little, if any, self-renewal, very little or if any. So they cannot just divide and renew themselves. They have lost that ability to, renew themselves. Okay, that is why they are destroyed after, for example, red blood cells after 120 days, they are destroyed. Yeah. Now, so these precursor cells are the blast cells, and their children or their offspring, or what they eventually get, which are the red cells, the granulocyte, the monocyte, the lymphocyte, and the platelet. So remember that the hematopoiesis begins from the stem cells, the totipotent cells, which give rise to the multipotent cells. Multipotent cells give rise to the progenitor cells. The progenitor cells, which are the colony forming unit, give rise to the precursor cells, which are the blast cells that now eventually give rise to the effector cells, which are red cells, granulocytes, and the rest of them. Okay, so now let's look at the mature effector cells. So the mature effector cells are the final product of hematopoiesis. They are the red blood cells, which carry oxygen, carbon dioxide, and then they have a lifespan of 120 days, which we already discussed before. And then the neutrophils, give rise the function in phagocytosis and the killing of bacteria and other microorganisms that get into the body. And then we'll have the monocytes, which also have phagocytic function, phagocytosis and killing of microorganisms. And then they help with what we call antigen presentation, which we would look at when we look at immune reaction. We'll talk about antigen presentation later. And then we have the lymphocytes, which identify foreign bodies. They act as our immune system, the main immune system, adaptive immune system, where we have the B lymphocyte and the T lymphocyte, and then we'll have the helper T lymphocyte, the suppressor T lymphocyte, and the killer T lymphocyte. And then we have the platelets, which help in hemostasis. to maintain normal blood volume and to prevent excessive blood loss during bleeding. So these are the mature effector cells. So you must not forget that before you get a red blood cell, we already first had a cell that was a stem cell, which was a totipotent cell. And that totipotent cell now gave rise to a multipotent cell, which now had a colony forming unit, GEMM, and then that colony forming unit now gave rise to a precursor cell, which are the blast cells, and then the blast, which are the blast, and then the blast now give rise to the cells, which are the red cells. That is the process. So now let's look at the diagram to explain everything that we've been saying. Okay, so this diagram here is going to show you how each of the blood cells are produced. Everything we've been saying is in this diagram. So let's start from here. This diagram began with the multipotent progenitor hematopoietic stem cell. So these are the stem cells. We said they are totipotent and multipotent. So here we have a multipotent progenitor stem cell. And this multipotent stem cell, we said, can give rise to a myeloid progenitor and a lymphoid progenitor. So this one here, myeloid progenitor, now will now split again. So what are the progenitor cells we get from the stem cells? We have the granulocyte monocyte progenitor. So this will be a CFUGM, for example, CFUGM progenitor cell. And then we also have the eosinophil basophil progenitor cell, right? That is with the granulocyte also, this forming granulocyte alone without monocyte. And then we have the megakaryocyte erythrocyte progenitor. So this will form platelets and red blood cells. So this is the myeloid progenitor. So if anything happens to this myeloid lineage, we would have a problem with granulocyte and monocyte, a problem with eosinophil and basophil, a problem with platelets and erythrocytes. But we will not have a problem with lymphocyte because lymphocytes are coming from a different line, a different multipotent progenitor cell. Then let's finish this side first. Then this granulocyte monocyte progenitor, from here you can see that it can give rise to what we call the myeloblasts, which are the precursor cells now, right? Myeloblasts and monoblasts. So myeloblasts will give rise to promyelocytes, myelocytes, metamyelocytes, band cells, and segmented neutrophils. So the final product will be the neutrophil, the segmented neutrophil. Before you have the segmented neutrophil, you had these stages of precursor cells. So from myeloblast, we didn't jump straight to neutrophil. It will still divide again to, or differentiate to promyelocytes, myelocytes, metamyelocytes, band cells, and segmented neutrophil. So the normal neutrophil you should find in your blood in high amount is this segmented neutrophil. Okay, so if there is a problem and differentiation stops here at this point, for example, if differentiation stops here at this point, what you will find in your blood is that there will be no segmented neutrophil, or there will be a decreased number of segmented neutrophil, but you will have a lot of promyelocytes and myeloblasts. Why? Because differentiation did not continue. It was arrested somewhere here. So if there is an arrest of differentiation or of hematopoiesis at this point, it means that metamyelocytes will not be produced, band will not be produced, and segmented neutrophils will not be produced. So in the blood, you will only find promyelocytes in high amounts, and you will find myeloblasts in high amounts. And that should show you, you know, I said when you find blast in cells, that is already a sign of leukemia. It means that there is leukemia, there is cancer of the blood, of the myeloblasts. Then, to form monocytes, the monoblast, which is the granulocyte monocyte progenitor cell, give rise to monoblast, and then the monoblast now give rise to what we call promonocyte, and this promonocyte give rise to monocyte. So you find monocyte in the blood. These are the final effector cells, the final products. Then let's look at the eosinophil and the basophil. Now, these myeloblasts will give rise to promyelocytes, eosinophilic myelocytes, eosinophilic metamyelocytes, eosinophilic band, and then you have eosinophil, similar to what you had with neutrophil. But this time around, it is eosinophil. Then for myeloblasts, for basophil, you have the myeloblasts will give rise to promyelocytes, basophilic myelocytes, basophilic metamyelocytes, basophilic band, and then basophil. So basophil, eosinophil, and neutrophil have almost similar method of similar stages of differentiation. Now let's move over to the platelets and the erythrocyte. So we have the megakaryocyte erythrocyte colony forming unit, which will now give us a pronormoblast. Pronormoblast will give us a basophilic normoblast. And then the basophilic normoblast will differentiate to form polychromatic normoblast. And the polychromatic normoblast will form orthochromatic normoblast. And then we'll have a polychromatic erythrocyte, which is also called a reticulocyte. Okay? For polychromatic erythrocyte is called reticulocyte. And the reticulocyte eventually gives rise to erythrocyte. What you should find in your blood is erythrocyte. When you find excess reticulocyte in your blood, and that should tell you that there is something wrong with the differentiation process. Instead of you seeing erythrocyte, you are now seeing reticulocyte. So it means that something happened at this point, and the reticulocyte could not completely mature into erythrocytes. And then megakaryoblast here will give rise to promegakaryocytes, and then megakaryocytes, and then megakaryocytes will give up fragments, which are now called platelets. The platelets are fragments of megakaryocytes, and then we'll have the platelets. And then for the lymphocytes, the common lymphoid progenitor will give rise to what we call the dendritic cell, the pre-B cells, and the pre-T, you could call them progenitors, and the natural killer cell. Now, this pre-B will give rise to what we call the B lymphoblasts. And the lymphoblasts will not differentiate and give rise to the B lymphocytes. If you find lymphoblast in the blood, then there is leukemia, either an acute leukemia or a chronic leukemia, depending on the number of lymphoblasts that you see. And then the pre-T cell will give rise to the T lymphoblast, and then the T lymphoblast will give rise to the T lymphocyte. So this is how hematopoiesis occurs. So now we have all the final cells: neutrophil, monocyte, eosinophil, basophil, erythrocyte, platelets, and lymphocyte. All of them have been produced, but they had to pass through different stages, from totipotent to multipotent cells to progenitor cells to precursor cells, and then till before you get the final effector cells or the final blood cell. And this whole process is very important because whatever happens to any of them will affect the blood, and it will reflect in the blood. If I have a problem, if somebody has a problem with this lymphoid lineage, it means that we cannot produce B lymphocyte and T lymphocyte. And that means we'll have a very poor immune system, and we'll be prone to all kinds of diseases, because any bacteria or any virus can just come into our bodies and we would suffer from it if something happens and we are not able to produce any of these cells. So anything that happens to hematopoiesis will affect us badly. Good. So here, let's look at specific differentiation pathways. It's a kind of repetition of what we already said. So now looking at neutrophils, the process will be granulopoiesis, because we said a neutrophil is a granulocyte. So the process of production of neutrophil will be a granulopoiesis. So the totipotent stem cell, we said, are the CD434 plus cluster of differentiation. And then we have the myeloid, they'll give rise to the myeloid stem cells. The myeloid stem cells will give rise to the progenitor cells, which are the colony forming unit, granulocyte, yeah, and the monocyte. And then we have the progenitor cells, colony forming unit, granulocyte alone. Now not with monocyte, but only granulocyte. And then we have the precursor cells, which are the myeloblasts, promyelocyte, myelocyte, metalomyelocyte, band cells, and then the segmented neutrophil, which is the final product. The kinetics of neutrophil production is 14 days from myeloblast to neutrophil. So from myeloblast till the formation of neutrophil takes 14 days. Takes 14 days. Okay, so this is diagram here, just showing you the whole process. So this diagram is like lifting one pathway, just one pathway from this place. Like lifting this pathway from this whole diagram. So that's what we have here. Common myeloid progenitor, and then myelocyte, and then you have the segmented cells. For red blood cells, we have the totipotent erythropoiesis, totipotent stem cell, CD434 plus. Then we have the myeloid stem cell, the colony forming unit. So here we have the progenitor, CFUME, CFUME progenitor. So these are a kind of blast forming unit. And then we have the precursor cells, proerythroblast, basophilic erythroblast, polychromatophilic erythroblast, orthochromatophilic erythroblast, reticulocyte, and then you have the final erythrocyte. And from the kinetics, from erythroblast to reticulocyte takes 5 days. Okay, it takes 5 days. The next one, the lymphocyte. So we have the antigen-independent phase. So lymphocyte production occurs in 3 phases. The antigen-independent phase, which there is initial lymphopoiesis that takes place in the bone marrow, and then maturation takes place in the lymph node. So the initial to form lymphocyte, the initial process takes place in the bone marrow, but the maturation of the cells take place in the lymph nodes and in the thymus gland. Now, the T lymphocyte will mature in the thymus gland, and then we have the antigen-dependent phase. So this is second cycle of differentiation and proliferation, and this happens only when you are exposed to an antigen. So when you are exposed to an antigen, for example, you are exposed to a virus, right? Your lymphocyte, there will be differentiation and proliferation of the lymphocyte. And the lymphocytes needed to kill the virus is the killer T cells. So there will be differentiation to form that specific T cell, right, as a result of antigen exposure. And then we'll have the memory phase. So the memory phase follows antigen exposure. When you're exposed also to an antigen, there will be differentiation. And when the T cells differentiate, they give rise to what we call memory cells. So they produce some cells to fight, and they produce memory cells. And these memory cells are able to easily recognize that antigen the next time you are exposed to that antigen. So the next time you are exposed to that antigen, there is no need for a new differentiation, because there is already a memory cell that can easily recognize that, oh, we've seen this virus before. It will be easier to fight and destroy that virus. And then for the platelets, we have the pro-megakaryoblast, and then we have the megakaryoblast, the DNA replication without cell division, and then we have the megakaryocyte, and then we have the platelet, which is the final product of fragmentation of megakaryocytes. So these are the growth factors we already mentioned before. Erythropoietin is synthesized in the kidney. induces proliferation of colony-forming unit erythrocytes. Synthesis rises when red cell level falls. So anytime there is decreased level of red blood cells or there's anemia, erythropoietin synthesis will increase so that there can be production of more erythrocytes. Then we have the stem cell factor, which stimulates totipotent, pluripotent stem cells to enter differentiation pathway. Then we also have granulocyte monocyte colony stimulating factor. These ones are secreted by lymphocytes, endothelial cells, stromal cells in the bone marrow, and they stimulate production of granulocyte and monocyte, as we said in the previous slide. And then they are available in large amounts. Then we have the granulocyte colony stimulating factor, which is secreted in the bone marrow, mainly but not exclusively stimulate the production and function of granulocytes. The synthesis rise in response to infection. So anytime you have an infection, you need your granulocytes to come and fight. You need your neutrophils to come and fight. So these are stimulated or produced. And then we have what we call interleukins, which we would look at later when we start talking about inflammatory reaction and immune reactions. We'll look more at interleukin 1, interleukin 2, interleukin 3, and all the interleukins and what each of them do in the process of immune reaction and inflammatory reaction. So they mediate multiple highly complex communications between various classes of white blood cells, and their level increases when there is a response to infections also. So the formation of blood cells, all the various types of blood cells are produced in the bone marrow. They arrive from a single type of cell, which is called the pluripotent stem cell, which is what we already said. And then these stem cells are very rare, only about 1 in 10,000 bone marrow cells. And they are attached probably by adherent junctions. So osteoblasts, so they are attached to bone precursor cells also lining the bone cavity. And they express a surface protein, which we already said is called CD434+. And they produce by a process called mitosis. They divide, and then they form two kinds of progeny. They can form more stem cells and cells that begin to differentiate along the paths leading to, so they form multipotent stem cells, and they form themselves, so they can renew themselves, and then they can give rise to multipotent stem cells. Then we have interleukin 7, is a major cytokine in stimulating bone marrow stem cells to start down the path leading to the various lymphocytes. So for lymphocytes to be produced, it is interleukin 7 that stimulates that. Erythropoietin will say stimulate red blood cell production produced by the kidney. Thrombopoietin, assisted by interleukin 11, stimulates production of megakaryocytes, and their fragmentation produces platelets. And then the GM-CSF, the colony stimulating factor for granulocyte and monocyte, and they lead to the production of granulocyte and monocyte, as we already said. Then they differentiate into neutrophil, and then interleukin 5 develop into eosinophils. Now pay attention to these. Interleukin 5 develop into eosinophil. We'll talk about this later when we're talking about allergic reactions and the role of interleukins. Interleukin 3 participates in differentiation to form basophil, and then macrophages are formed from the macrophage colony stimulating factor. So this is everything you need to know about the hematopoiesis. It's a kind of complex topic, but you need to know it because it's very important. In all exams that you're going to do, you will find questions, all professional exams, you'll find questions about hematopoiesis, questions about leukemia, questions about progenitor cells. You're always going to find questions like this, and they will help you diagnose any problem with red blood cells or white blood cells or platelets, be able to diagnose them properly. So that will be all for hematopoiesis. You would do well to watch this lecture again, this video again, to try to remember everything and study it very well, because Yeah, there's a lot of information here, but if you can just remember this diagram and understand the diagram, then it will be easy for you to remember stem cells, progenitor cells, precursor cells, and then effector cells. And then you just pick each cell and see how they are produced. Okay, is there any question?
No question, sir.
No question. Okay, so that will be all for today. We'll see you in the next lecture. Thank you.
Thanks.